Hello and welcome to the 2021 Embedded Vision Summit. My name is Patrick Bungert, and I serve as the Vice President for Artificial Intelligence at Samsung SDS, which is the IT company in Samsung Group. Today, I want to tell you about a highly data-efficient deep learning approach to train an AI model on visual data, on images, and particularly our aim is going to be in the medical image domain, where we want to detect and diagnose or localize a particular disease. And we're going to be focusing on COVID-19. First of all, we're all familiar, of course, with COVID-19 by now. And sometimes you get certain symptoms that might be indicative of COVID-19 or some other diseases like flu or pneumonia. And you don't really know whether you have COVID or not. If you're feeling just moderately ill, maybe you can afford to wait a certain amount of time before the nasal swab test comes back from the lab after a day or two. But sometimes people are in an acute condition, they have trouble breathing, they need to go on the ventilator, receive medication, and in that case, it is time sensitive to diagnose them with either COVID or something else so that you can give them the proper medication. Now, the laboratory test, as I said, requires 1 or 2 days to come back from the laboratory. Some people in acute states maybe don't have that amount of time available. Plus, the test kit is only 94% accurate, so you're not 100% sure. There's a rapid test kit available, which gives you an answer much more quickly, but it's less accurate than that. And so, again, you have a certain risk attached to that. Also, you are relying on whatever locale you're in, actually having these tests and the chemical laboratories available to actually make use of that test. And not every location does. An X-ray machine is a lot more ubiquitous around the world, and taking an X-ray, looking at an X-ray is a stable process that is very common even in other parts of the world. And so the question is, can we, in an acute patient, detect COVID based on a lung X-ray? And of course, the answer is yes, otherwise I wouldn't be here today. So we're going to talk about how to actually make that happen. Now, using artificial intelligence to do anything requires a data set that is made up of both the input data, in this case, X-ray images, and the desired output data so that we can do machine learning in order to train the AI on the correlation between the inputs and the outputs. In other words, we have to label each image with the information of whether this particular lung is a COVID-infested lung or not. And labeling these medical images is a problematic task. So first of all, there are certain issues of legal difficulties. The images are protected by HIPAA legislation in the United States, which restricts access to the data. Of course, it is also expensive to obtain the data. You actually have to get a large number of individuals to an X-ray and take that image and collect it centrally somewhere. And then knowledgeable people, in other words, doctors, have to go in, look at each one of these images and attach the appropriate classifier to that. In this particular case, we have over 15,000 different people's chest X-ray available. And so 15,000 images had to be opened by an expert doctor and labeled. In fact, every image has been labeled by 6 different doctors. So 15,000 images were each labeled 6 times in order to get a democracy kind of voting regime on which image is which class. And so it's really quite costly to come up with a labeled data set like that. And so an associated question is, can we get away with using less data? So we're all familiar with the slogan big data, where usually the answer to all problems is get more data. But in cases like this, medical imaging, where acquiring more data is difficult or expensive or just not possible, we have to think in terms of the slogan small data and see what we can do with the restricted amount of data that we actually have. And so here we're also going to be using a technique called active learning to try to label the right images, the most informative images first, so that we can cut down on the amount of data that we need. So in this particular case, those 15,000 odd images were from 3 different categories. There were lungs that are normal, no disease at all. There were lungs that were infected by pneumonia, which is a disease that presents similar symptoms to COVID. And then there is COVID, which of course is again a lung infestation, but it's much more deadly. So we had many more normal cases than the average. We had more pneumonia cases, and we had relatively few COVID examples, but it's COVID that we're interested in because that's the important disease. And so the first thing to do for an AI system, you don't want to overrepresent one category, otherwise a model that says it's this category is going to have a pretty good accuracy already. You need to oversample the category that you have fewer data points on and undersample the category that you have lots of data from. So undersampling, of course, is very easy. You randomly pick a certain number of examples from a large data set. In oversampling, you have choices on how to do that. Many people add noise, for example, to create a slightly different image. In this case, lung X-rays are not particularly diverse, and so we did not add noise or anything of the kind. The oversampling was done in the most trivial ways. In other words, the same image was chosen more than once without any modification. In that way, we chose the same number of normal pneumonia and COVID images to train the AI system. But prior to training, the images were registered. That's something that is essential for image data sets, particularly those that are focused on a particular object. So when you're doing a chest X-ray, for example, you have on that image certain bones like the collarbone or the shoulders that are also on that image, and they're not relevant to detecting the kind of mucus distribution in the lungs that you're really interested in to detect these diseases. And you don't want some kind of spurious correlation between the shape of the collarbone and COVID to matter. And so you have to cut that out. So there's a pre-processing step where you shrink down the image from the whole chest X-ray to an X-ray of only the lung area and remove everything else so that you're focusing on the right parts of the image. Next for this is a kind of feature learning. So we're used to feature engineering being a technique that is primarily used in tabular data analysis. So where we're talking about numerical data arranged in a table or a time series. And then features are like the columns in that table. And I can create additional columns. That's feature generation, for example, by taking the ratio of 2 existing columns. And then I do a feature selection, in other words, choosing which of the columns are informative about my data set, and going on to then use these selected features to train my model. Now, in deep learning or computer vision, we actually have to go through a feature engineering step also, except that the features are no longer columns in a table, but the features are of a different nature. So in this particular example here, we are using sub-images as potential features. In other words, if you have a larger image, you might consider a particular cutout, let's say this little portion over here, as a feature of the image. And now it becomes a matter of deciding how big is a feature, where in the image is a feature typically located, and those questions lead to feature engineering in the case of computer vision. And so being a part of an image becomes a metric in the space of images. It becomes a kind of similarity measure. And this process then allows us to sort and cluster images into groups of images based on those features in that high dimensional space. This can be made more formal in a technique called SimClear. There are 2 references in the slides that will provide you with all the mathematics and algorithmics behind that. And ultimately, this allows us to propel classification of computer vision images to a better standard than the state of the art prior to that kind of feature engineering. So you have the image, then you have the SimClear layer, and then we add one fully connected neural network layer at the back of that, which converts the vector representation of the image that's output by SimClear and outputs out of the fully connected layer one of the 3 possible class labels that we're desiring here. So then the idea of active learning is to say we want to cut down on the number of human labels by inquiring the human labeler to provide only a few labels, but we, the active learner, get to decide which images the user is supposed to label. So the idea is to sort the images in order of information gain. We begin with an initial a step, unsupervised feature learning, that provides the initial batch of images to label. Those are then labeled by the human labeler, provided back to the active learner. The active learner then learns an ordering over the space of all images, sorts them, provides the user back the next most informative batch of images. Those are labeled, provided back, and then starts a back and forth conversation in which the user labels a few images and the active learner asks for more images to be labeled. What you observe in this is that the overall accuracy of an artificial intelligence model trained on only the labeled images at any one time, at any one batch, rises logarithmically like we see in the orange line on the screen here. And it hits, roughly speaking, the maximum achievable accuracy after approximately 16% of the entire data set having been labeled. Now, 16% is obviously a piece of information that's relative only to the specific data set here. It might sometimes be less or might sometimes be a little more, but nonetheless, it is relatively little number of images that have been labeled compared to labeling everything. The blue curve on the same image is the accuracy that you would have achieved if you would have labeled the images in a random order. And we see that there is a haphazard up and down as we would expect early on. There's a general trend for the accuracy to go up, of course, but you can see that in order for the model accuracy of random order labeling to hit the kind of heights that we get with active learning, we need to label close to everything in the data set. And so the difference between labeling in a random order and labeling in an information gain order are very, very significant in terms of human labor. Of course, we're talking about the amount of human labor that has to go into it. At the end of the day, of course, we need to label the entire data set anyway. The only question is how much is labeled by human and how much is labeled by AI. In addition to that, we have to ask ourselves, how fast is all of this processing going on? So if we're talking about active learning. Every day I label a batch of images, and then the active learner has to, of course, learn from that. So there's a certain processing time that needs to happen. And we use distributed training to make that fast. Distributed training means I distribute my training over multiple computers, not using one GPU, but I might use many GPUs in order to do a single training task. Now, we're used to that not being linear, it being sublinear in the CPU processing, because there is overhead. And so some of the processing goes towards processing the overhead, and then the remainder can go to processing the actual workload. Now, in the case of GPU processing, that turns out fortuitously not to be the case. As you use more GPUs, the overall processing actually gets faster. There's a superlinear scaling to this. And the ultimate reason for that, simply put, is that the amount of memory available is per GPU. In other words, as you double the number of GPUs, you also double the amount of working memory. And therefore, you can increase the batch size, not the batch size for labeling, but the batch size for training the active learning model. And therefore it runs faster. So you can actually improve the training time and basically an arbitrary amount just by scaling up the amount of resources. So I like to talk, instead of about real time, I like to talk about realistic time. So realistic time for me is either lunchtime, you can run it in less than an hour. It's dinner time, you can run it between the end of one working day and the beginning of the next, or weekend, you can run it between Friday night and Monday morning. Those are the three time scales that are really relevant. And in this particular case, you can easily use a few GPUs together to crunch the active learning step into a lunchtime, meaning that you can label 2 batches a day quite readily, and therefore step through this process quickly. The state-of-the-art accuracy for this particular data set of 15,000 odd lung X-ray images is 91%. We can match that accuracy after only 16% of that data is labeled. Of course, the correct 16%, the most informative 16%. Having done that, the active learner is capable of automatically labeling the rest of the images, the other 84%, and then the human doctors only have to validate those labels. Now, validating labels is always easier and faster, significantly, than producing labels of your own accord, and therefore, again, you get this huge labor saving. And having that full data set labeled allows the accuracy to go up from 91% to 95% on test data. And that is an accuracy that's not only the highest accuracy to date in observing COVID-19, but it is actually higher than the accuracy of the physical test that you get if you put a nasal swab in your nose. So there's a real a kind of stepping stone here where the AI jumps from being less accurate than the chemical test to being more accurate than the chemical test. So there's an evolutionary step in this methodology. Ultimately, this is our confusion matrix on the right, as compared to the confusion matrix of the most accurate paper to date prior to our study on the left. And you can see that the recognition efficiency of detecting true COVID-19 cases as COVID-19 is 95%. And some COVID-19 cases, 3% are detected as normal, and 2% are detected as pneumonia. That is, of course, to be expected. You always get a certain number of false positives and false negatives in any AI system. But we can see that the overall accuracy has gotten better by this methodology, which again, to sum up, included multiple objects. We had feature engineering in terms of SimClear plus a fully connected layer to do the full training of the entire labeled data set. But we also demonstrated that using ActiveLearning through the process I described allows us to save a significant amount of human labor. And in this case, of course, we did both together. In other words, we were able to significantly lower the amount of time and cost required to make the model. And at the same time, we were able to make a model that's 4% more accurate than the state of the art today. So put both together, it's a better model for less investment using this technology. Ultimately, of course, then we can say that in combination, the methodology of doing feature engineering for deep learning and using active learning to obtain the labeled data set is a combination of techniques that allows you to be much more effective and efficient with artificial intelligence. And the combination with distributed training allows the whole thing to be done not in very long computational timescales, but essentially over that proverbial lunchtime so that you can get things done pretty quickly and put your models out there to practical use. Ultimately, in this case, coming up with a model for COVID-19 that is even more accurate than the physical test. If you would like to read more information about this, I put together some papers for you. On the left-hand side, you see a scientific article at the top if you're interested in the full machine learning details. If you're more interested in the popular ramifications of this, there are two LinkedIn posts below that go into some detail on this. And lastly, there is a video that demonstrates the technology from A to Z that we use to do this. On the right-hand side here, you have 5 links to data sets that are the chest X-ray images that we used. So the 15,000 odd images I was talking about is the unification of these 5 data sets there, which are all free of charge and open source, so you can download them and do your own study about them. So thank you very much for your attention. You're welcome to reach out to me by email or on LinkedIn if you have any questions after the event. And for now, I'm happy to answer your questions live. Thank you very much.